Hello, my name is Troy McComak, and today my presentation is on the kidneys, more specifically the glomerular filtration rates and the forces that are involved with the kidneys. So first, let's just review the kidney here. We know that the kidney is an organ in the body. We have two of them, and they're located in our abdomen, in the back towards the spine. And what the kidney does is it's a filter of blood. And the way it works is it has all these little parts inside of it. You can see here's a cross section of the kidney. And there's tubules, and they're called nephrons. And then um, inside these nephrons is the glomerulus, which is where the blood comes in. And we can see it here. And that's where the blood comes in. And the urine comes out through the tubule here. And you can see the urine goes out that way. I showed my daughter a picture of this. She's only 4, and she says it looked like an orange. So I just decided to kind of put that picture in perspective there of kind of like an orange, and that may be similar kind of how it's structured on the little parts of the kidney there. So the glomerulus is a very important aspect of the kidney. And what it is a small intertwined group of capillaries within the nephrons of the kidney that filter the blood to make the urine. There's hydrostatic pressure, which is the force pushing and exerted on the blood vessel. It is the primary force that drives glomerular filtration. So we're going to be talking a lot about these other forces that are at play within the glomerulus. So here we have a good diagram of kidney function. Now, there's two parts to the glomerulus. We have the Bowman's capsule, and that's where the urine is excreted out into, and we have the glomerular capillaries, and that's for the blood. And you can see the blood comes in to the afferent arterioles. Pressures are reacted in the glomerular capillaries. The blood is filtered in a process called renal ultrafiltration, renal meaning kidney. The force of the hydrostatic pressure in the glomerulus is, from the blood itself, is really what's driving the force of these filters, the filtration there. And then we go and the blood comes out to the efferent arteriole, and there's going to be some reabsorption, and there's also going to be secretion that for telling the kidneys and the urinary tract how to respond, maybe from some hormones. And then the blood comes out to the renal vein after having been filtered out of these poisonous compounds like urea. And it gets filtered out into excretion. And so there's filtration minus reabsorption plus secretion is excretion. So we can kind of see there's a lot of arithmetic that goes involved with, how the kidneys are working. So there's a lot of different pressures. There's osmotic pressure, also called oncotic pressure or colloid osmotic pressure. And how that works is it's induced by proteins, notably albumin, which is the protein that's like egg whites. And it's in the blood vessels that are surrounding in this glomerular capillary. And what it does is it allows water to be removed, and with that, some also filtration elements like urea. So here's another look at glomerular filtration. It's caused by the force of the difference between hydrostatic and osmotic pressure through the glomerular filtration rates, includes other variables as well. And we can see here we have these purple arrows, which is the glomerular blood and the hydrostatic pressure of the blood. And then you have the blood colloid osmotic pressure reacting on the blood vessels, as well as the capsular hydrostatic pressure reacting. And what's going to happen is, in this situation, you have a 10 millimeters of mercury being the net pressure that's outwardly into Bowman's capsule there. Here's another look at the glomerulus, and here we can see more of a calculation being done where we have a 55 millimeter Hg of glomerular blood hydrostatic pressure. The capsular hydrostatic pressure is at a 15 millimeters of mercury, and we have the blood colloid autosomatic pressure at 30 millimeters of mercury here. And it's all reacting in 3D very quickly. This is a chemical reaction, so it's happening very fast. And urine is being made right here in the kidneys, and it goes right out through the proximal convoluted tubule here. And we can see the calculation of the GBHP, which is the number 1 here, the glomerular blood hydrostatic pressure, 55 minus 15 of the CHP, and then minus the 30, which gives us a net of 10. So in normal day-to-day function, the rate of glomerular filtration is determined by kidney perfusion. However, changes to the composition of blood or obstruction of the urinary tract can influence the rate of filtration. For filtration to occur at all, the pressure of blood in the glomerular capillaries must exceed 2 other forces at play. These are the osmotic force, the proteins that retain within the capillary that they exert, and the pressure of the fluid within Bowman's capsules. And so here we see a different designation, which is the pressure of the glomerular capillary. Here is given 60. The pi GC, which is the osmotic force due to plasma protein, and the pressure in Bowman's capsule, which is just a reiteration of what we already went over before. So glomerular filtration occurs because the blood pressure is really just higher, and than that of the osmotic pool of the proteins within the capillary, and the pressures of filtrate within Bowman's capsule. In the example here, if the PGC fell to 45 millimeters of mercury, no filtration would occur. In different, they give slightly different estimates out there of how these can occur, but everything's really within about 5 millimeters of mercury of pressure. So this is a pretty textbook standard here. So what's actually going on in the glomerulus there? Well, here's a look at the glomerular basement membrane. So kind of a more zoomed in view of the kidney here. Here we have Bowman's space, which is where the urine is concentrated. And here we have the capillary lumen, which is where the blood's at. So here we have the endothelial cells, the glycocalyx. This is where it's all occurring, and you have the fenestrae here, which is our openings. And these this glomerular capillary wall consists primarily of fenestrated endothelium, the basement membrane, and epithelial foot processes. The foot processes form filtration slits spanned by slit diaphragms. Also shown here is the endothelial cell coat, which is this glycocalyx. Some approximate dimensions are, 8 to 10 nanometers, but in between the fenestra, it can be 30 nanometers. And we could also see that there's these epithelial podocytes, which occur on the outside and allow for the movement of filtrate. There's also these in these epithelial podocytes, there's a space of about 40 nanometers. So this brings us to our next topic, and that's problems within the kidneys. In a normal uh, glomerulus, here we could see, it keeps the red blood cells, white blood cells, and most proteins in the blood, and only lets out the watery fluid into the urine. However, um, sometimes those can be a little leaky, those endothelials, and there can be some breakage there, a diseased portion of the kidney there. And so what happens is we have proteinuria and hematuria, meaning protein in the urine and blood in the urine. And you can see here, as those break open, red blood cells come out, proteins come out, and those can be detected using modern medical practices. And we're going to be looking into how those are treated and what does that really mean. So there's a lot of issues that can cause the glomerulus to break down um, and the glomerular filtration rate to decrease, which can ultimately lead to big problems. It means the kidneys aren't functioning. So the kidneys are shutting down, or there's a system problem called chronic kidney disease. In this study, they found that the glomerular filtration rate and albuminuria for detection and staging of acute and chronic kidney disease in adults was based on that if the confirmation of albuminuria, meaning albumin in the urine, is required because of, there's differences during the day of the variation of these conditions, which affects, creatinine excretion and muscle masses or, diet. Um, that the albumin excretion rate should be measured from a timed urine collection. So it needs to be measured, over a certain period of time in order to find out, what's going on. And they also found that the detection and staging of acute and chronic kidney disease uh, is relatively simple because of the morbidity and mortality associated with kidney disease. Early diagnosis is important and should be pursued in at-risk populations. So we should be screening a lot for this because once the kidney damage is done, it's hard to fix unless there's going to be dialysis or a kidney transplant. Pretty much, we don't want to damage our kidneys, and we need to be checking for this type of problem during routine checkups, and quite often in order to stop the kidneys from being at risk of damage. So once the kidneys are damaged, there's not much that can be done. I mentioned there's dialysis that can occur. That's pretty much a mechanical filtration rather than having the biological filtration, and it needs to be done quite often. And also the transplantation. But before we get there, this study here was pretty cool because it looks at the maintenance of the having pretty much maintenance hemodialysis, or having these dialysis all the time, is often prescribed 3 times a week. And they found there that there's a less frequent type of dialysis that can be done, not just 3 times a week, as long as they're able to look at the residual kidney function, or the RKF. And it's important to look at residual kidney function because even though the majority of the kidney doesn't work, there's still aspects of it that do work that can help so that they don't need so much. So and because what's going on is, if there's the blood is being filtered by an outside mechanism, all these forces at play are going to kind of shut the kidneys down. And so they want to keep that residual kidney function quite at a high rate because it's going to, they found that the kidneys would shut down if they just kept doing dialysis. And mainly, looking at survivability of the patient, and they notice that there's a higher mortality after the first year of dialysis in those with the lowest residual kidney function. So trying to keep patients alive for the longest is the general outcome. Well, I thank you for your time. Here are the references I've used in my presentation.